The year is 1966. In a small office at Boston University, the air is thick with the scent of old paper, chalk dust, and the quiet hum of intellectual ambition. A young biologist, her mind a whirlwind of data and defiant insight, makes the final corrections on a manuscript. Her name is Lynn Margulis, and the document she holds is titled On the Origin of Mitosing Cells. It is not merely a paper. It is a declaration of war against the established order of evolutionary biology. For decades, the story of life had been told through a single dominant lens, neo-Darwinism. This was a world of slow, gradual change where random mutations were sifted by the relentless sieve of natural selection. Life was a brutal, competitive struggle, a red in tooth and claw affair where organisms battled for supremacy. The cell, the fundamental unit of this struggle, was seen as a unified, singular entity, its internal components, its organelles, having evolved slowly and methodically from within. This was the dogma, repeated in every textbook, taught in every lecture hall, and defended fiercely by the titans of the field. Lynn Margulis, however, saw something entirely different. Peering through the lenses of microscopes and at the scattered data points others had dismissed, she saw a history of life that was not just about competition, but about radical, revolutionary cooperation. Her claim, laid out with meticulous detail in her manuscript, was breathtakingly audacious. She proposed that the most critical components of our own cells were not truly ours at all. The mitochondria, those tiny powerhouses that burn fuel to give us energy, and the chloroplasts, the green engines that power plants through photosynthesis, were, she argued, the descendants of ancient free-living bacteria. Billions of years ago, she theorized, a larger primitive cell had engulfed these smaller bacteria. But instead of being digested, a truce was called. The smaller organism found a safe harbor, and the larger host gained a powerful new ability to either generate immense energy from oxygen or to harness the power of the sun. It was a merger, an acquisition, a permanent alliance forged in the crucible of early life. She called this process symbiogenesis, and it suggested that the greatest leaps in evolution weren't gradual steps, but sudden, transformative unions. This was not just a new chapter in the story of life. It was a completely different book, written in a language the establishment refused to understand. She typed the final word, stacked the pages, and began the arduous process of submission. She sent her revolutionary idea to the most prestigious scientific journals in the world, the gatekeepers of accepted knowledge. And then the replies began to arrive. They came in thin, university-branded envelopes, each one a paper-thin dagger to her theory. One after another, they landed on her desk. The rejections were not just polite disagreements, they were dismissive, even contemptuous. "Your work is too speculative," wrote one editor. "This is not a subject for a serious scientific journal," declared another. The criticisms piled up, forming a wall of institutional resistance. Her theory was labeled far-fetched and unpublishable fantasy. Some reviewers didn't even bother to hide their scorn, calling her ideas the work of a crackpot. In total, she would receive a stack of 15 rejection letters. 15 times, the door to scientific discourse was slammed in her face. The world of biology, a field supposedly dedicated to uncovering the truth about life, had looked at one of its most profound secrets and collectively decided it was insanity. The system had spoken. It had deemed her a heretic. But the evidence, hidden inside nearly every cell on the planet, was waiting patiently for its voice to be heard. The question was no longer whether she was right, but whether a system built on dogma could ever afford to admit she was. Following that initial, brutal dismissal, a lesser scientist might have abandoned the theory, filed it away as a youthful folly, and moved on to more acceptable research. But Lynn Margulis was not a lesser scientist. Fueled by a certainty that bordered on obsession, she refused to be silenced by the chorus of rejection. If the establishment wouldn't listen to her hypothesis, she would force them to confront the evidence. She doubled down, refining her arguments, gathering more data, and finally, in 1967, her paper found a home in the less prestigious but more open-minded Aura. This publication became the beachhead from which she would launch her intellectual assault on the fortress of neo-Darwinian orthodoxy. Her strategy was simple: to connect the dots that other researchers were observing in isolation, weaving them into a narrative so compelling it could no longer be ignored. Her first line of attack was visual. The development of powerful electron micrographs allowed scientists to see the cell's interior with unprecedented clarity. Where others saw simple, unified organelles, Margulis saw relics of a forgotten independence. She pointed to the membranes. A cell's own components typically have a single membrane, yet both mitochondria and chloroplasts had two. This, she argued, was the smoking gun of an ancient engulfment. The inner membrane, with its unique chemical composition, was the original bacterial cell wall. The outer membrane was the remnant of the vesicle, the bubble of the host cell's own membrane that had wrapped around the bacterium as it was swallowed. It was an anatomical ghost story, a tale of invasion and integration written in lipids and proteins. Furthermore, she highlighted that these organelles reproduced on their own schedule, dividing by binary fission, just like free-living bacteria, completely independent of the cell's own mitotic cycle. They were tenants living in a cellular apartment, following their own rules. From anatomy, she moved to the core of life itself, genetics. The central dogma of the 1960s was absolute. A eukaryotic cell's genetic blueprint, its DNA, resided exclusively within the nucleus. To suggest otherwise was heresy. Yet, Margulis pointed to a growing body of strange, inconvenient findings. Researchers had discovered that both mitochondria and chloroplasts contained their own DNA. And this DNA wasn't the long linear chromosomes found in the host cell's nucleus. It was a small circular loop of genetic code, strikingly, undeniably similar to the chromosomes found in bacteria. It was like finding a separate, secret instruction manual inside your television, written in a different language for a completely different device. This genetic anomaly was a devastating blow to the theory of gradual internal evolution. How could a cell's own components evolve their own separate and distinctly foreign genetic system? The only logical explanation was Margulis's. They were never the cell's own components to begin with. Her final piece of evidence was biochemical, a functional proof of her theory. She compiled data on the effects of antibiotics. Drugs like streptomycin and tetracycline were known to be lethal to bacteria because they targeted their specific type of ribosomes, the molecular machines that build proteins. When these same antibiotics were applied to eukaryotic cells, a strange thing happened. The host cell was largely unharmed, but its mitochondria and chloroplasts stopped working. Their ribosomes, it turned out, were bacterial in nature, just like their DNA and their membranes. The very weapons designed to kill bacteria were also crippling these essential organelles. The evidence was no longer just circumstantial. It was a clear biochemical fingerprint linking the organelles to a bacterial past. The data was overwhelming, whispering its truth from every corner of cell biology. Yet the resistance remained fierce, rooted not just in scientific disagreement, but in a deep-seated cultural bias. The academic gatekeepers, overwhelmingly male and invested in the competitive gradualist model, found her theory of symbiotic leaps too sudden, too cooperative, perhaps too feminine for their worldview. The resistance was systemic. But with each new piece of data, the fortress of dogma began to show cracks. how much longer could the establishment afford to ignore the revolution happening from within every living cell? Building on that relentless pursuit of truth, the whispers of evidence she had gathered began to echo in labs across the world, growing into a roar that could no longer be ignored. For nearly 2 decades, her endosymbiotic theory remained on the fringes, a controversial but compelling idea. The final, irrefutable proof would come not from a microscope, but from a revolution in technology that Margulis herself could have only dreamed of in 1966. The advent of rapid DNA sequencing and phylogenetics in the late 1970s and 1980s gave scientists the power to read the book of life letter by letter. For the first time, they could directly compare the genetic code of the organelles with the vast library of free-living bacteria. This was the ultimate test. If Margulis was wrong, the DNA of mitochondria and chloroplasts would show some distant, ancient relationship to the cell's nuclear DNA. If she was right, their genes would be a near-perfect match for specific types of modern bacteria. The results, when they came, were breathtaking. They were the final, undeniable vindication of her life's work. The genetic sequence of mitochondrial DNA was not just like bacterial DNA, it was a clear and direct descendant of a group of microbes called alpha proteobacteria, which includes the modern-day Rickettsia genus. The case of the chloroplast was just as conclusive. Its DNA was a direct match for cyanobacteria, the free-living photosynthetic organisms that oxygenated our planet's early atmosphere. The genetic data was a fossil record more reliable than any stone, proving beyond any doubt that our cells are chimeras, composite beings forged from an ancient, world-altering alliance. The debate was over. The heretic had become a prophet. The once scorned theory of symbiosis was now the endosymbiotic theory, a foundational pillar of modern biology. Textbooks were rewritten, lecture notes were updated. An entire generation of biologists was now taught that the origin of complex life, the very evolutionary leap that led to fungi, plants, animals, and us, was not a product of slow, plodding change, but of a radical act of cooperation. Lynn Margulis, once the pariah who received 15 rejection letters, was finally brought into the fold. The accolades that had been denied to her for so long began to pour in. In 1983, she was elected to the prestigious National Academy of Sciences. In 1999, President Bill Clinton awarded her the National Medal of Science, the highest scientific honor in the United States, for her outstanding contribution to the understanding of the living world. Her name was finally etched into the pantheon of great evolutionary thinkers along Darwin. But the recognition, however welcome, could not erase the decades of struggle, the dismissal, the ridicule. The story of her fight serves as a powerful and often uncomfortable reminder of the inertia of scientific dogma and the systemic barriers that face those who dare to challenge it. Her legacy is not just the theory itself, but the intellectual courage it took to defend it against a world that called her insane for seeing a truth hidden in plain sight. It compels us to ask a crucial question. How many other profound insights are we missing right now? How many brilliant ideas are being dismissed as too speculative? How many cures are being delayed? How many truths are being silenced simply because the person proposing them doesn't fit the mold, or because their discovery is too disruptive to our comfortable view of the world? The universe holds its truths. Our only job is to be brave enough to see them.